To summarize what we said on the previous lecture is that the quicker a cell turns over, going from labile to quiescent to non-mitotic, the longer the S phase. This is true not only for regular cells, but it's also true for tumor cells. And that's why if you ever get a path report where it reports that the tumor has a high or large or long or big S phase, that's not a good sign. It's very hard to talk about stem cells these days without offending somebody's religion. Nevertheless, I'm going to try. There's basically two types of stem cells. There's embryonic and adult. And often the word totipotential is used to describe stem cells, sometimes multipotential or pluripotential or omnipotential. But the result is the concept of a stem cell is a cell that is capable of producing a whole wide variety, perhaps all types of tissues. And in the example of a fertilized ovum, that is a truly omnipotential stem cell, isn't it? Because as we know from basic biology, it gives rise to all types of human cells. Let's talk about different roles of stem cells. They're so necessary in studying differentiation of cells because, as you know, stem cells are the ultimate undifferentiated cell. And as they differentiate through various generations and mitoses, perhaps under the influence of cytokines and growth factors, they then become more differentiated, which we defined as losing some of its capability but gaining specialization, just like in specializing from general medicine to pathology. In research, we can knock out genes in stem cells and then see what diseases these mice will have, and it's basically been a tremendous help in how we learn about diseases. As when I was in medical school back in the Stone Ages, before they had cars, or when we walked around with clubs and bows and arrows, there was always talk about repopulating damaged tissues with cells that contained better genes. Well, folks, they're still talking about it. Are we that much closer to actually doing it? I don't know, but that's the ultimate goal with stem cells, and someday that may be achieved. In the adult, the bone marrow has cells. They were formerly called hemocytoblasts. They still are sometimes, which are capable of differentiating into all the different types of marrow cells. Sometimes a very small percentage of these come out in the peripheral blood as well, and that's basically the basis for stem cell harvesting, where you take somebody's stem cells, grow them, and then give it back to them, perhaps after you've already knocked out their bone marrow with chemo or radiation. It's been a very, very valuable therapy in medicine. In normal tissue development, you can see relatively totipotential cells in normal places in which they differentiate the growth centers, like the base of a hair follicle, like you see here. And the principle is, the more undifferentiated the cell is, the more it can give rise to more differentiated structures under conditions of what we're talking about, regeneration and healing. Here is a part of a liver, which doesn't look terribly exciting, but in where the arrows are point to cells which basically give rise to normal hepatocytes. These little stem cells in adults are all over, and they're not truly stem cells. For example, these cells here at the limbus cannot give rise to all types of cells, but it certainly can give rise to all types of cells in that area with regeneration. So here's the basic principle. You have a cell, is probably going to look like the most boring cell in the world. Let's use the bone marrow as an example. Or perhaps it may look very, very boring. Why? Because it doesn't have any differentiated appearance. Perhaps we want to call it a pluripotential cell. Under the influence of growth factors, cytokines, and the so important extracellular matrix, it can differentiate into several different types of connective tissue cells, or even epithelial cells if it was really, really a totipotential cells. So here's your pluripotent stromal cell in the marrow under the influence of MyoD, myogenin, and others, differentiates into a myocyte. Under the influence of vascular endothelial growth factor or fibroblast growth factor 2, might differentiate it into an endothelial cell. Under the influence of SOX9, it's going to become chondroblasts and chondrocytes eventually. Under the influence of CBFA1, osteoblasts. Under the influence of PPAR gamma, fat cells. So that's the whole concept of stem cells or primitive cells differentiating. Under the influence of growth factors and cytokines, these boring looking nothing cells differentiate. And in the process, once again, they lose some of their totipotentiality, but they gain specialization. And that's the whole process of growth, isn't it? Let's go back to our basic trio in embryology called ectoderm, mesoderm, and entoderm. Well, the reason why mesoderm is brown and the other two are blue is because mesoderm basically gives rise to all of the tissues that we call stroma or connective tissue, pretty much what we saw right here. Whereas ectoderm and entoderm give rise to epithelial cells. Ectoderm, perhaps epithelial cells of the skin and neuroectoderm, which is brain, spinal cord, and entoderm, which is epithelial cells of the GI and respiratory tract. So the principle is, you take your primitive 3 embryonic layers, and we can see a pattern of differentiation from these layers into very specific types of tissues. And think about it, isn't this another example of differentiation? And is the blastocyst ultimately derived from a fertilized ovum? So there we are, folks, going from one cell to every single specialized type of cell you could think of in various stages under various controls of cytokines and growth factors to become what we call adult differentiated tissues. And that's going to lead us into our next big topic of growth factors, the GFs. And I'm going to just give you the introduction, and then we'll go through all of them in the next group. There's a lot of different growth factors. They all end with the word GF. And they do a lot of things, but as you might guess, they cause cells to grow. They cause cells to multiply. They are mitogenic, all of them. But what they do in general, before we get into the specific ones, is that they are involved in locomotion of cells, contractility of cells, this marvelous concept of differentiation of cells, and in the case of healing and regeneration, and especially healing, growth of new blood vessels, which we call angiogenesis, or organizing inflammation, or granulation tissue. They're all polypeptides, and if you want, you could all call them cytokines too. So let's keep it at that, and we'll go to the specific ones in the next group. Thank you very much.